Hello everyone, my name is Manu, and today I'm going to introduce my project, which is called Genestorian, an open source web application for model organism collections. So first, let me give a bit of background on the project. In a genetics lab, we typically work with recombinant DNA, and this is DNA that is outside of cells. We typically have it in a test tube. And the good thing about recombinant DNA is that we can take existing molecules. So for example, here we have two circular DNA molecules, which we call plasmids. And so we can take these two plasmids and generate a new plasmid that contains sequences from the existing ones. And there's different ways of doing this. One of them is using restriction enzymes. So we can cut open this plasmid and make it linear. And we can also cut the piece of this plasmid that we are interested in, and then we can combine them to generate a new circular plasmid that contains the piece of this one that we were interested in. In the lab, we are also interested in working with model organism strains. I, for example, work in yeast. And what we typically do is we modify the genome of these organisms. So, for example, here I've taken a fragment from a plasmid and I have introduced it into the genome of this yeast. So by this, I have generated a new version of a gene, which I call an allele, but I also generated a new version of the organism that I call a strain. And what typically one can do also with many organisms is to cross them sexually. So we can take two strains that contain two different alleles, and among the offspring, we will obtain one strain that has both. So as you can see, when working with strains, we have to handle both the generation of alleles, which resembles the generation of recombinant DNA because it's basically combining existing sequences to generate new ones, but also the inheritance of these alleles that exist in strains. So we have to track two lineages, the lineages of the sequences of recombinant DNA, but also the lineages of alleles in the strains. And what is the problem? The problem is that tracking recombinant DNA lineage is possible with proprietary software, but we cannot export this information in a way that is readable by computers. And for strains, there's just simply no software that does this, and people just tend to use Excel. So of course, it's very inconsistent, and the information about laboratory collections cannot be used or read by computers. So the goal is to develop an open standard to describe how existing entities are combined to generate new ones, and from this make an open source web application where you can have a laboratory inventory where all the sequence can be traced through cloning steps up to the entry point into the collection. So during